So my talk today is going to be on the role of the endothelial glycocalyx, which I'm really excited to hear has come up twice today already. But this talk is not going to be about its role in COVID or in its role with chitinase-like proteins. This is going to be about the role of the glycocalyx in skin inflammation. So hopefully this is a figure which is relatively familiar to you all. This is the process of leukocyte transmigration, and it's how immune cells will get out of your blood vessels and into the tissue, or in my case, the skin. We know that leukocytes, to begin with, will form weak transient adhesions with the endothelial adhesion cells, which will cause them to roll across the surface until they gradually slow down and will eventually form firm adhesions before coming to a stop altogether and passing through the endothelium. This is a really interesting, well-characterized process. However, there are certain layers that the immune cell has to pass through which are less well-characterized. And the one which I'm going to be talking to you about today is the endothelial glycocalyx. So, the glycocalyx is a thick layer of glycoproteins and glycolipids, and it's found on the surface of all mammalian cells in some form. But it's particularly prevalent here on the lumen of the blood vessel. You can see it forms like a furry layer that can be up to 2 microns thick. And in this way, it actually protrudes out further into the lumen of the blood vessels than those endothelial cell adhesion molecules, which I mentioned before, were so important to leukocyte transmigration. In this way, the glycocalyx has been shown to have some form of regulation over leukocyte migration. It has some pro-adhesive effects. Firstly, it can actually mediate some of the initial interactions between leukocytes and the endothelium. In fact, heparan sulfate, which is an important component of the glycocalyx, can bind to L-selectins on leukocytes and mediate some of these important initial interactions which can mediate rolling. It can also tether chemokines to the endothelium via its glycosaminoglycan side chains. And this allows the formation of a chemokine gradient, which can kind of tell us immune cells whereabouts they should be migrating out of the blood vessel and into the tissue. However, as it is such a thick layer, the glycocalyx has also been shown to have some anti-adhesive effects in the tissue. It can actually act as a barrier between leukocytes and those adhesion molecules and prevent immune cell migration into the tissue. In many types of inflammation, it's actually been shown that the glycocalyx can be degraded and remodeled, which will expose those endothelial cell adhesion molecules and lead to increased immune cell transmigration into the tissue. I'm looking at the glycocalyx in psoriasis, which is a chronic inflammatory skin disease which affects around 125 million people worldwide. I'm looking at plaque psoriasis, which you can see from this kind of painful image on the right, and this affects the vast majority of patients. Psoriasis has a mix of genetic and environmental causes, but it has a very strong genetic basis, and around 36 of the susceptibility loci for psoriasis are actually immune-related genes. If we look at the histopathology of psoriasis, there are 2 main hallmarks which I'm particularly interested in. The first being this epidermal thickening. You can see this purple epidermis is far, far thicker in the skin of psoriasis patients than it is in healthy patients. And the second hallmark, and this is due to the hyperproliferation of keratinocytes or skin cells. The second hallmark, which I'm interested in, is this infiltration of immune cells into the dermis. And this is mediated by the IL-23, IL-17 cytokine axis. And a particularly important cell type in early inflammation in psoriasis are neutrophils. I use a mouse model of psoriasis for my experiments, Aldara cream. I've shown it here how it looks on the dorsal skin of a mouse, but I actually apply it to the mouse ears. I just thought this gave a bit of a better image of how it emulates the human disease in its clinical signs. The active ingredient in Aldara cream is imiquimod, which is a TLR7 agonist, and it induces this psoriasis-like skin inflammation by that same IL-23, IL-17 pathway as in the human disease. And again, neutrophils are an important cell type in this mouse model of the disease as they are in the human disease. So my hypothesis is that given that this immune cell migration into the dermis is such an important part of the pathology of psoriasis, and given that they're going to have to pass through that endothelial glycocalyx in order to get here, my hypothesis is that the endothelial glycocalyx undergoes changes in inflammatory conditions, such as with Aldara cream, and these facilitate the entry of cells into the tissue. And if we dysregulate this glycocalyx, this will have some effect on the outcomes of psoriasis-like skin inflammation. So this is more of a structural diagram of what the glycocalyx looks like. And I'm just going to draw your attention in particular to heparan sulfate. And this is a highly sulfated glycosaminoglycan, which is really integral to some of the roles that the glycocalyx has. I've shown here that heparan sulfate is indeed expressed on endothelial cells in Aldara cream-treated mice. You can see I've stained for heparan sulfate here in green and CD31 in red, and that's a marker for endothelial cells. And you can see some co-localization here of both the CD31 marker and the heparan sulfate, which suggests that there is indeed an endothelial glycocalyx present in Aldara-treated mouse skin. I'm interested in, however, into what happens when the glycocalyx is modified. So I introduce you to the heparanase enzyme, and this is the only enzyme which we know can cleave off those heparan sulfate side chains of the glycocalyx. It can also lead to further modifications of the glycocalyx, as when it cleaves off those side chains, it leaves the core protein more exposed to cleavage from other enzymes. And it's actually been shown in vitro that if you add heparanase to an endothelial cell monolayer, that it's associated with increased adhesion and rolling of immune cells. So from our collaborator in New Zealand, we got hold of a heparanase enzyme inhibitor, and I pre-treated my mice with this for 1 day by intraperitoneal injection, and then every day for 6 days alongside the topical application of Aldara cream before culling the mice on day 8. So my hypothesis for this experiment was that under normal conditions, the glycocalyx can form a thick, intact barrier which will prevent immune cell migration into the tissue. However, under inflammatory conditions, that heparanase enzyme would come along, cleave off the heparan sulfate side chains, deplete the glycocalyx, exposing adhesion molecules, and more immune cell transmigration would occur. However, I thought when I added the heparanase inhibitor, it would lead to a more intact glycocalyx, and this would act as a barrier against immune cell migration. So inhibiting the heparanase enzyme would lead to a more intact glycocalyx, which would be a thicker barrier against immune cell migration, and I thought this would lead to less inflammation. However, this is actually not what I found at all. I found that when I inhibited the heparanase enzyme, so that's the pink line here, the clinical signs of inflammation in psoriasis were actually worse than the PBS and Aldara group in purple. We have 3 readouts for inflammation in psoriasis in this particular model, so ear thickness, ear redness, and ear scaling. And all 3 readouts were significantly worse with the heparanase inhibitor than in the PBS and Aldara control. And because I was so surprised by this, we repeated this 3 times, and the same trend happened across all 3 experiments. So to work out a little bit what was going on here, I ran flow on the skin of these mice to see what was happening to the immune cells. And I found that actually, despite the increased psoriasis severity, the number and proportion of CD45 positive immune cells are reduced when the heparanase enzyme is inhibited compared to that Aldara and PBS control, despite the fact that the number of CD45 negative cells, so your non-immune cells, was unaffected. To have a little closer look at what was going on here, I looked at individual immune cell subsets, and I found that different types of T cells show that same trend. There's a reduction in their number with the heparanase inhibitor compared to control. The same can be said for macrophages, DCs, and monocytes, and B cells also show that same trend of a reduction in number with the inhibitor. I mentioned before that neutrophils are a very important cell type in psoriasis, and they're particularly interesting to my project as they're very migratory. You can see in the flow plots alone that there aren't many neutrophils in just PBS-treated mice, but when you add Aldara cream, there's a big influx of these cells into the skin. However, when you add the heparanase inhibitor, again, this is that significant reduction of neutrophils into the skin in both number and proportions. However, a really interesting finding from this experiment was that monocytes, which in theory should behave in a similar way to neutrophils, they're also very migratory, they're an early responder to inflammation, they did not actually show this same change. So even though neutrophils did show a change and most of the cell types did, monocytes seem to be unaffected by this inhibitor, which is quite a surprise. I sort of expected a blanket effect of this inhibitor on all the cell types. But it does seem that inhibiting heparanase selectively affects certain cell types and not others. As to why the clinical signs of inflammation were worse, but even though immune cell migration was lowered in the skin, in my most recent repeat I had FOXP3 as a marker for regulatory immune cells, and I found that these cells were reduced in number and proportion when we added the heparanase inhibitor. And perhaps this could provide some kind of explanation as to why, even though immune cell migration to the skin was lower, the inflammation was worse. Maybe they're having like a disproportionate effect on how much inflammation we see in the skin. So for now, that's sort of where my experiment is at the minute. We're currently working on profiling this and working out a bit better what's going on in this process. But just to conclude, we found that inhibiting heparanase can increase the severity of psoriasis-like skin inflammation, but it can reduce immune cell infiltration into the skin, but it doesn't seem to affect monocytes in particular. This suggests that inhibiting the remodeling of the glycocalyx using these inhibitors in the skin can selectively affect the recruitment of certain immune cell subsets and not others. As to why we have increased severity but decreased migration of immune cells, maybe it's that fewer regulatory cells getting into the skin can cause increased skin inflammation, particularly when a lot of our IL-17 producing immune cells, like ILC3s, gamma delta T cells, are largely skin resident, so will be unaffected by changes in migration. That's it for me for now. So I want to thank everyone in the Saunders lab, the Dyer lab, and the Lennon lab, and to our collaborator in New Zealand, Olga Zupkova, and to Shafkat's drone footage, giving us a pretty sick video to finish our presentation. So thank you.
was another really clear talk, and I'll just point out to the PIs, all the students kept the time pretty much. So, any questions for Megan?
Really nice talk. So in your model, the heparin sulfate was just on the endothelial layer. Is there any heparin sulfate on the actual immune cells, and could that be playing a role?
Yeah, so immune cells will have their own, sorry, did I interrupt you there? Immune cells will have their own glycocalyx, and they will have heparin sulfate on this surface as well. But just in that one I was showing it is on the endothelial cells.
So when you're blocking the, when you're using the heparanase, the enzyme inhibitor, could it be that as well as...
That will affect both, yeah. So both the glycocalyx on the immune cells and the glycocalyx on the endothelium will be affected, yeah.
And could that be, you factor that into
that could also
model?
be, yeah, and there is also heparin sulfate as part of the basement membrane in the endothelium. It's like present in a lot of places, so it might not just be the endothelial glycocalyx on the, this, but I am doing some IHC at the minute to sort of see A, where heparanase is, and B, where heparin sulfate is, to see what's going on a bit better.
Hello? Ooh. Yeah, that was fantastic, but just three things come to mind. So firstly, heparin sulfate is really big, and the way that you presented it was that well, if you get rid of it, everything will bind. But really, when it's there and when you get rid of it, you're changing the size of receptors and ligands that preferentially interact, not just making them all bind or not bind. Essentially, the big receptors and ligands could still bind each other when heparin sulfate is there on both the cells, but now you're allowing the smaller proteins to interact. But the bigger ones, like a lot of the inhibitory ones, would still interact. Secondly, the protrusions that come from cells would overcome any, so maybe you're allowing more interactions happening through the protrusions only. And then finally, I think NKp46 binds heparin sulfate, which is on NK cells, and that activates. So have you looked at the NK, you put all these other cells, but ignored the best one?
Yeah, so to try and cycle back through your 3 points there, so the glycocalyx, also a point I didn't mention, has a negative charge, which will be matched by, so the glycocalyx on the cells will also be repulsed by the glycocalyx on the endothelium. So that does act as a bit of a buffer as well. So things like mainly adhesion molecules, so the glycocalyx can extend like 2 microns into the lumen, whereas some of the like ICAM selectins like 30, 40 nanometers into the lumen. So it is quite, like it goes way, way further. As for inhibitory like receptors, I'm not too sure about that. And also NK cells, I did have a look at them, but my staining wasn't brilliant, so I didn't want to put it on here and make a bold statement about how they were when I wasn't that confident in my staining. That answers everything.
Hi, Megan. Really nice talk. This is Claude. I was just wondering, because do you know if the heparanase inhibitor alone does something for like, is like has cytotoxic activity or something like that, and that would explain why you have like more damage?
That is something we've thought about. I think we are going to repeat this again and do a group that might just have the inhibitor and nothing else. There is another group, I think, I'm not sure where, but in Bristol that have done this in diabetes, and I think they did do a heparanase only group. As for like what effects I think I might think it might have, it's actually a heparin sulfate mimetic. So hopefully it wouldn't have a cytotoxic effect, because it should be quite structurally similar to heparin sulfate. But yeah, no, we haven't had that as a group yet, so that's a very good point. Thank you.
You're welcome.
Any other final questions?
Hi, yeah.
Jonathan?
You were saying about the Tregs and the fact that you think that the Tregs were sort of disproportionately, well, there was an imbalance perhaps where the Tregs were disproportionately not present. Mm-hmm. So don't you have absolute numbers? Can you not assess that?
So I think I might have phrased that. I mean, not the disproportionate numbers, so I do have the cell numbers on the slide. It's that perhaps even though all immune cells, fewer of them are getting into the skin, maybe the Tregs are having more of a, they're not disproportionate in number, more the effect that they have, like not getting as many in the skin is having a big effect on the amount of inflammation, if that makes sense.
Mm. And what about the difference between, so you didn't see a decrease in the non-immune cells. Mm-hmm. So what do you think is going on there between those two subsets? So immune cells versus non-immune cells, the fact that there's a difference there, do you think that, is there anything there that's going on that's driving that phenotype?
So hopefully that would sort of back up my point in that, so the CD45 negative cells, that will be things like the keratinocytes, the skin cells, I think fibroblasts. So if they're not changing a number, it does suggest this is a migration thing, and this is the immune cells migrating into the skin with the Eldora cream, and there being a difference with the inhibitor, if that answers your question.
Okay, I think we've had some good discussion. We need to keep to time, so thanks Megan, and thanks to all of the PhD students for keeping to time and giving lovely talks.